Welcome everyone to this week's edition of the Extracellular Vesicle Club. I'm really excited to have another opportunity for the genitourinary system EV's special interest group of ICEV to host this event. And so we have Dylan and Elena here who are going to help with the moderation, who are going to do the moderation. And this is a very nice collaboration. So this is our first special interest group, or SIG, that was established under the ICEV umbrella. And they've been very active. Many of you have probably participated in some of their events, like the recent Education Day that they held virtually. And they will also be holding an event that Dylan was just telling me about at the annual meeting of ICEV, the International Society for EVs. So this is a meeting that's held in Vienna. It will start next Wednesday, so just a week from now in Vienna, Austria. And if you have not signed up and you're somewhere in the neighborhood, I would definitely recommend going there. You can go to the Education Day alone on Wednesday, or you can go for the whole meeting from Wednesday until Sunday. And we have a very large number of registrants, so 1,700 so far. It's probably going to even bump up a little bit by the time we get to the meeting. So hope to see many of you there in any case. So I'd like to hand this over now to Dylan and Elena to introduce our special speaker for today.
Yeah, thank you, Ken. And you couldn't encourage people to attend the ICEV meeting more. It's a fantastic meeting. There'll be great opportunities to engage with GUSEV and the other SIGs. So this particular session is quite timely. Obviously, we have ICEV 2025 in Vienna coming up, but the other reason this session is so timely is that a little under a month ago, this manuscript titled "Circulating Extracellular Vesicles as Putative Mediators of Cardiovascular Disease in Pediatric Chronic Kidney Disease" was published. And we're really lucky today to have one of the senior authors on that study, Dr. Szandor Simmons, here to share their work. So Szandor, welcome.
Yeah, thank you very much for the kind introduction, and especially to make it possible that I present our recent study on extracellular vesicles in pediatric chronic kidney disease. So what we actually aimed here is to clarify whether extracellular vesicles in chronic kidney disease may contribute to vascular dysfunction, and especially therefore also then for cardiovascular disease development. So chronic kidney disease, or short CKD, with its prevalence of around 11% in adults and an incidence of 12 per million of the age-related children population, is a disease characterized by progressive loss of kidney function and decrease in glomerular filtration, which then ultimately leads to accumulation of so-called uremic solutes or uremic toxins. In most of us, and so I assume that most of you are healthy individuals, these toxins are excreted by the kidney, but in chronic kidney disease patients, they are associated with cardiovascular morbidity and mortality. In fact, the leading cause of death in chronic kidney disease patients is cardiovascular disease, yet the mechanisms that cause cardiovascular disease in chronic kidney disease are not fully understood. The high cardiovascular mortality in CKD patients is thought to be largely driven by cardiovascular risk factors such as hypertension and diabetes, especially in adult CKD patients. Therefore, most pediatric CKD patients face increased cardiovascular morbidity, leading to cardiovascular death as their primary cause of mortality. But what mediators other than uremic toxins are released due to local renal dysfunction and can spread systemically, contributing to cardiovascular disease and its complications? And here, extracellular vesicles come into play. You all know very well that extracellular vesicles are lipid bilayer delimited particles comprising, for example, 30 to 150 nanometer sized exosomes and 100 to 1,000 nanometer sized microparticles that are naturally released from almost all types of cells. They carry a cargo of proteins, nucleic acids such as microRNAs, but also lipids, metabolites, and even organoids from the parent cell, and therefore are considered to play an important role in intercellular and interorgan communication in both physiological processes and onset and progression of disease. Microparticles are directly formed by outward budding and fission of the plasma membrane of the host cell, and thus contain receptors and other cell surface molecules which enable the identification of the cellular origin and allow it to be distinguished from microparticles from other cells. It has previously been demonstrated that EVs have been changed in number and composition in chronic kidney disease. So here we explore mechanisms of EV release in CKD and hypothesize that both uremic toxin exposure and circumstances of extracorporeal kidney replacement therapy are causative for the release of extracellular vesicles that promote endothelial damage and cardiovascular disease development. Children with CKD are indeed a valuable study population for understanding cardiovascular disease development and identifying kidney-specific disease mechanisms free from common lifestyle-related factors. We performed a cross-sectional study comparing 94 individuals with end-stage kidney disease without and with kidney replacement therapies such as peritoneal dialysis, hemodialysis, as well as kidney transplantation in relation to an age-matched healthy donor group. Furthermore, we were lucky to investigate the consequences of kidney transplantation in 12 individuals in a longitudinal follow-up. Now, the average age of our cohort was 10.9 years, and 49% of the participants were females. The estimated glomerular filtration rate, or short eGFR, is a key indicator of kidney function and helps in the diagnosis and monitoring of kidney disease. A normal eGFR value is generally considered to be 90 or higher, but values as low as 60 are considered normal if there is no other evidence of kidney damage. Our healthy control group has an eGFR of about 150, while end-stage disease is characterized by an eGFR of 32 in CKD stage G3 to G5. And an eGFR of about 7 to 11 in those who have received extracorporeal renal replacement therapy. Patients after kidney transplantation showed stable graft function with mean eGFR of 104.5 that corresponds to the eGFR of the healthy control group. Longitudinal follow-up after kidney transplantation was performed in 12 CKD patients during the study period, allowing for intra-individual comparisons of EV parameters with impaired and restored kidney function. In this cohort, the mean time after kidney transplantation was actually 5 weeks, within the kidney function normalized to a mean eGFR of 94.5 in these patients. All groups of CKD patients exhibited marked cardiovascular morbidity as displayed by arterial hypertension and left ventricular hypertrophy. Moreover, resistant hypertension that remains uncontrolled despite using multiple blood pressure medications, including a diuretic, was more common in CKD, peritoneal dialysis, and hemodialysis patients than in patients after kidney transplantation. To investigate the impact of extracellular vesicles in the pathophysiology of chronic kidney disease, we isolated extracellular vesicles from plasma of the respective groups of our cohort and confirmed effective isolation by negative staining transmission electron microscopy and immunoblots for CD9. Nanoparticle tracking analysis confirmed that mean EV size was unaltered among all donors and patients. Now, NTA further show that peritoneal dialysis patients have higher extracellular vesicle concentrations as compared to healthy donors and kidney transplant patients. Notably, EV concentration did not significantly change within individuals following kidney transplantation, despite restored kidney function. To analyze changes in circulating hematopoietic and endothelial cell-derived extracellular vesicles, multiparametric flow cytometry was applied to cell-free plasma to account for 8 cell lineages, including the most prominent hematopoietic lineages and endothelial cell lineage. Platelet-derived extracellular vesicles were the most abundant EV fraction. However, as with B-cell-derived EVs, monocyte EVs, neutrophils EVs, and erythrocyte EVs, there was no difference between patient groups and healthy donors. Cross-sectional analysis confirmed a threefold increase in concentration of endothelial cell extracellular vesicles of hemodialysis patients as compared to kidney transplant recipients. In addition, concentration of macrophage-derived EVs, but also T-cell-derived EVs, were increased in chronic kidney disease patients without dialysis in comparison to healthy donors. In longitudinal comparisons of chronic kidney disease patients before and after kidney transplantation, we only observed significant decrease in endothelial cell-derived extracellular vesicles. And this fact specifically caught our attention as endothelial cell extracellular vesicles have been linked to cardiovascular disease in adults, and dialysis patients carry the highest cardiovascular risk among all chronic kidney disease patients. To assess a potential functional relevance of CKD extracellular vesicles, we first analyzed the EV sphingolipid content as an EV cargo with potential functional relevance in cardiovascular disease. Using lipidomics, no consistent changes, however, were observed in CKD and hemodialysis patients compared to healthy controls, with the exception of an upregulation of C16 sphingomyelin in chronic kidney disease patients, but not in patients that received hemodialysis. We therefore focused on microRNAs, which have been shown to be abundant in extracellular vesicles and involved in post-transcriptional gene regulation target cells. Through microRNA sequencing on isolated plasma EVs and partial least squares discriminant analysis, we observed a distinct separation between individuals with normal kidney function, so that means healthy donors and kidney transplant recipients, and dialysis patients who received either peritoneal dialysis or hemodialysis, and chronic kidney disease patients that lay in between. Bioinformatic analysis revealed 31 microRNAs that significantly differed between the CKD peritoneal dialysis and hemodialysis group and the healthy and kidney transplant recipient group. Next, we identified cellular target processes of the altered microRNAs in chronic kidney disease EVs. Gene set enrichment of these genes identified enriched GO terms that include several cellular processes associated with vascular homeostasis, which are dysregulated in cardiovascular disease pathophysiology, such as cellular response to PGGF stimulus, regulation of smooth muscle cell proliferation, or regulation of angiogenesis or angiogenesis by itself. For real-time PCR validation, the dysregulated microRNAs were sorted by the probability to interact with genes of the enriched GO terms, resulting in the selection of 5 microRNAs. And among them, microRNA 24-3P, 103A-3P, and 142-3P were cross-sectionally reduced in peritoneal dialysis and hemodialysis patients compared to healthy donors with restored levels after kidney transplantation, whereas LET7D5P and microRNA 19A3P were higher in kidney transplant patients compared to peritoneal dialysis and hemodialysis patients. Furthermore, longitudinal analysis revealed significant increased levels of LED7D5P, microRNA 24-3P, 103A-3P, and 142-3P after kidney transplantation. So these results suggest that circulating extracellular vesicles in CKD patients contain lower levels of specific microRNAs compared to healthy individuals with potential functional relevance in cardiovascular disease processes and reversal of extracellular vesicle microRNA dysregulation after kidney transplantation. To assess the functional relevance of these EVs on the vasculature, we exposed primary human aortic endothelial cells to EVs from healthy donors, from hemodialysis patients and kidney transplant patients for 18 hours and performed bulk RNA sequencing from these cells after extracellular vesicle treatment. Among the 6 GO terms related to vascular homeostasis predicted by microRNA target identification, targeted gene analysis revealed patient group-specific clustering with the strongest dysregulation of genes belonging to the GO term angiogenesis. So this suggests the functional relevance of dysregulated EV microRNAs for altered expression of angiogenesis genes. Therefore, we focused on a functional analysis of angiogenesis and endothelial cell migration in vitro. So notably, HUVECs treated with extracellular vesicles from hemodialysis patients showed significantly reduced vascular tube formation compared to healthy extracellular vesicle-treated cells, resulting in a significantly increased mesh size but unaltered number of branching intervals. Furthermore, HUVECs treated with extracellular vesicles from hemodialysis patients showed impaired migration and impaired proliferation compared to extracellular vesicles treated cells. Of note, aortic smooth muscle cells, which I don't show here, did not show any comparable changes in proliferation. So therefore, extracellular vesicle microRNAs may be associated with functional extracellular vesicle effects on the vasculature, potentially driving phenotypic changes such as capillary rarefaction that are well described in chronic kidney disease. Finally, we aimed to elucidate potential mechanisms of extracellular vesicle release in chronic kidney disease. And actually, there is increasing evidence that microbiome-derived uremic toxins, particularly those resulting from tryptophan metabolism, such as indoxyl sulfate, are associated with cardiovascular disease and pediatric chronic kidney disease. So therefore, we hypothesized that tryptophan-derived uremic toxins may trigger extracellular vesicle release in chronic kidney disease. And so we analyzed metabolomics, or we performed metabolomics, and found that tryptophan metabolites discriminated between individuals with normal kidney function, so again, healthy controls, and kidney transplant patients, and CKD patients in a stage-dependent manner. When we looked closer on the species of the tryptophan metabolites, we realized that the balance of uremic toxin has shifted from tryptophan, shown here in green, and which are most abundant in healthy and individuals and patients who received kidney transplants, to indoxyl sulfate, shown here in dark red and most abundant in CKD and dialysis patients. So, in fact, formylkiniurenine and indoxyl sulfate and xanthorenic acid correlated positively with endothelial cell extracellular vesicle plasma concentration. And due to its prior cardiovascular disease association in chronic kidney disease, we chose indoxyl sulfate for endothelial cell stimulation in vitro. However, when we treated human aortic endothelial cells with increasing concentration of exogenous xanthorrhinic acid, formylkine-urenine, or indoxyl sulfate in vitro, we couldn't detect any effect on extracellular vesicle release. So, considering that endothelial cell EVs were increased in patients on hemodialysis, we investigated whether hemodialysis treatment affects the acute release of extracellular vesicles. To assess this, we recruited an adult patient cohort and estimated endothelial cell EV pre and post hemodialysis, which you can see here. Endothelial cell EVs did not change significantly after dialysis, suggesting that hemodialysis itself is not the trigger for extracellular vesicle release in these patients. However, a breakdown of endothelial cell EVs or EV concentration by vascular access chosen for hemodialysis revealed a 2.5-fold higher mean endothelial cell extracellular vesicle concentration in individuals with an arterious venous fistula compared to a central venous catheter. Of note, the vascular shear stress in an arteriovenous fistula is strong when the extracorporeal blood is returned into the vasculature. We therefore hypothesized that increased vascular shear stress acting on venous endothelial cells, either alone or in combination with tryptophan metabolites, may trigger endothelial cell extracellular vesicle release. To test this in vitro, we cultured human saphenous vascular endothelial cells under a venous shear stress with 1 dyne per square centimeter, or arterial shear stress with 10 dyne per square centimeter, and exposed them to vehicle control or indoxyl sulfate under constant flow for 18 hours. We specifically focused on indoxyl sulfate, as previous studies have suggested its role in vascular remodeling, and as indoxyl sulfate levels correlate with cardiovascular disease and pediatric chronic kidney disease. But high shear stress alone, as shown here, or indoxyl sulfate alone, did not induce additional EV formation. Yet in human saphenous vascular venous endothelial cells treated with high shear stress in combination with indoxyl sulfate, EV release increased compared to the lower shear stress control. Of the 5 microRNAs identified in our patients, 4 were detectable in the EVs released from the human saphenous vascular endothelial cells by high shear stress and indoxyl sulfate. Especially LET7D 5P microRNA 24RP 24-3P and microRNA 103A3P were reduced by high shear stress and indoxyl sulfate. Of note, the addition of indoxyl sulfate alone already affected the microRNA cargo, as shown by the significant reduction of LET7D 5P and microRNA 19A 3P. Therefore, we investigated whether extracellular vesicles from other cell types were similarly affected by indoxyl sulfate. You remember that I showed you at the beginning that we detected platelet EVs as the major fraction of circulating EVs and macrophage-derived EVs, which were already altered in their abundance in early stages of chronic kidney disease. So we focus on these cell types and analyze EV release and cargo after incubation with indoxyl sulfate. But as you can see here, similar to endothelial cell EVs, indoxyl sulfate alone did not result in increased extracellular vesicles in either platelets or macrophages. So in conclusion, in chronic kidney disease, impaired renal excretion changes balance in uremic toxins and vascular mechanical stress, leading to endothelial cell and immune cell activation with a subsequent release of extracellular vesicles, especially from endothelial cells. So we identified here several extracellular vesicle microRNAs as promising potential therapeutic targets that could be tested in future studies to restore impaired angiogenesis in chronic kidney disease. Yeah, and with this, I would like to come to an end, although it's still a bit time left. But anyhow, but I don't do this without thanking all the amazing people that contributed to the work, especially Felix Behrens, a former PhD student in my lab, who has pushed the study forward together with Johannes Holle, as a clinician scientist at the Department of Pediatric Gastroenterology and Nephrology and Metabolic Medicine at Charité University Hospital. And of course, also my fellow senior authors, Wolfgang Kübler and Henrik Bartholomeus. And of course, all the other persons who helped with the bioinformatic analysis, proteomics, lipidomics, and so far and so on. And I would like to thank you for your attention, and I'm happy to take any questions if you may have. Thank you.
Perfect. Thank you, Dr. Simmons. Fantastic presentation and really interesting data. While the questions are coming in, maybe I'll take the prerogative of asking the first question.
Sure.
And I'm wondering, and maybe you didn't have the numbers for this, but did you look at all at the impact of length of time on dialysis or time since transplantation on your results? In particular, I'm wondering if you had any patients since transplantation that was less than a year where the anti-rejection regimen would be quite different.
Yeah, unfortunately, we had not the chance to follow that up. But of course, it would be a very interesting aspect to look at. Yeah, but we unfortunately we didn't had the chance to follow up.
Perfect. Yeah. And then sort of related to that, the groups with the hemodialysis, the peritoneal dialysis, and transplantation, were there individuals that had been on multiple modalities in the study, or were they all sort of just one exposure?
They
So
had
could
just
you have
one.
been on PD and then transitioned to hemodialysis as an example?
No, actually, so we made sure that they had only one modality. So what we actually believe, so the accumulation of endothelial cell extracellular vesicles is maybe something that accumulates over time. Yeah, peritoneal dialysis patients are usually within a very young age. Yeah, and so they don't receive hemodialysis, or if they receive it at a later time point. And hemodialysis patients may have had the hemodialysis for several years. So we believe that the constant exposure to the shear stress caused by the hemodialysis may cause the accumulation of endothelial cell extracellular vesicles.
Okay. Perfect. Any question? Oh, we have one. Go ahead, Elena.
Yeah, there is a question in the chat. So, urine or plasma, which is a better source for EV to study the association between CKD and cardiovascular diseases?
Yeah. So I would so I think actually that so if you would like to go for pathomechanisms, that actually plasma extracellular vesicles maybe are the ones that do the trick here. However, we have currently a different story where we actually look on hyperaldosteronism, and what we see here is actually that extracellular vesicles may serve as markers for disease. Yeah, so I think urine extracellular vesicles are maybe very interesting to look for as a biomarker in disease progression. Yeah. However, we haven't analyzed in our CKD patients the urine extracellular vesicles, but it's definitively something that we would like to look at in the future.
Thank you. Are there other questions? Yes, there is one more. So interesting data, great presentation. The question is from Ole Alcurin, I hope to pronounce the name right. What % of all circulating extracellular vesicles are derived from endothelial cells?
So maybe let's go back to the data. So actually, here you see the total numbers, yeah. So in hemodialysis patients, yeah, we have around 8 times 10 to the 5 EVs per mil, while actually in the hemodialysis patients, this increases to around 2 times 10 to the 6 cells per mil. Yeah, so I think that's the increase that we see. I think the important point here is that actually the quality of the EVs changes. Yeah, so the cargo of the microRNAs completely changes. And so I don't think that the percentage maybe does, or the change in the percentage does here the trick. It's more that the microRNA cargo and the consequent post-transcriptional translation in the target cells actually makes the difference here.
Thank you. Can you guys hear me?
Yes, we can.
I was wondering more of, and you know, I'm a pediatric nephrologist, knowing very little about vesicles, but trying to learn. And I think that my understanding that they can be coming from endothelial cells, from immune cells, maybe from kidney cells, even that are injured, right?
Yes.
So, and you focused on endothelial cells, which makes sense in dialysis patients. Endothelium is so injured, right? Probably not only from shear stress, but other factors. But what % of all vesicles are coming from endothelial cells, as opposed to immune cells, like numerically?
So the percentage, actually, I don't know. We have here always calculated the total cells per mil. Yeah. So actually, I would have to look that up again. I don't remember exactly. But it was a significant number. That's the only thing that I can
because
tell
here,
you.
no, thank you. You like in A, you are showing EC-EV, right? Then MAC EV. So I'm assuming that you had markers, right? Like cell-specific markers
Sure. Yeah.
for each, and just like
yeah. So we have classical lineage markers like CD31 for endothelial cells, CD68 for macrophages, CD3 for T-cell derived extracellular vesicles. Yes. Yeah.
So at least the concentration seems similar, right? For endothelial derived, macrophage derived, and T-cell derived, 10 in 6, so something similar.
Yeah, absolutely. So I think so. We focused mainly on the endothelial cell derived extracellular vesicles because we see after kidney transplantation that actually the number dropped in individual comparisons. Yeah. Which actually macrophage EVs and T-cell EVs didn't do. But we don't exclude that immune cell-derived extracellular vesicles also contribute to the phenotype that we see. Yeah. So, we just saw that the qualitative changes in endothelial cell EVs in regards to the microRNAs is actually the most prominent one.
Yeah, well, I think
Thank you.
Most literature on this has them in the single-digit percentages, but not less than a percentage of the total. I think.
Yeah.
Okay.
It varies from study to study, but I think that's fairly consistent.
Got it. Thank you.
I have actually a question on my own. I'm from totally different field, but actually working on prostate cancer. So I was a bit surprised to see some of my usual bad guys in the microRNA list over there, like MIR21, MIR19,
Mhm.
7D, 142. I recognize those microRNAs.
Those,
So
yeah.
could you maybe just share your thoughts on that? Have you find any signatures that are overlapping with the oncogenic process, or are you talking general inflammation, or what is going on?
Yeah, so let me go back to the GO terms here. So actually, this is the one that we found, yeah. And so I don't know if there are some GO terms that you would consider as some that you also see. Yeah, I don't know.
The
G1 to
second
S.
one, maybe the G1 to
Yeah,
S transition. Yeah.
correct. Yeah. So I think there are several, but yeah, what it stood out for us were these effect on the vasculature itself. Yeah, and that's also one reason why we focus then on endothelial cell extracellular vesicles.
It makes sense actually, also because tumors, at least ours, have kind of pretty disturbed vasculature as well. So that could be
Yeah.
indeed.
Okay. Yeah.
Okay. Okay. Thank you so much.
Thank you.
This is Phil Askinase at Yale. Can you hear me?
Yes, we can.
Yes, I can hear you.
Hello, Dylan.
Hey,
And
Phil.
It's a fantastic study you've done and all very valid, but I come from another direction. EVs are very diverse,
Mhm.
and the changes of the subsets you're talking about is one approach. And I wonder if you have another approach of being able to give the isolated EV populations to mice and follow their renal function, or to some in vitro preparation of kidney. Because I'm very strong on the diversity, and then the numbers can be raised for many, many reasons and are not the crucial ones affecting renal function. So I'm very much in favor of following function, not numbers.
Yeah, so I completely agree with you. And actually, this is something we actually wanted to do, but we were limited by the amount of extracellular vesicles or plasma that we could receive from the donors because actually they were quite young. So sometimes we were just able to receive a milliliter of plasma. Yeah, sometimes we had patients who were 1 year or 2.5 years or something like this. So
Yeah.
we were simply limited by the total amount of extracellular vesicles. We considered, and I think this is something that we will follow up, to pool actually plasma from the different cohorts and then go into the animal model. Yeah. And of course, it would be very interesting to see if we see effects on the cardiovascular system after treatment with extracellular vesicles from healthy donors versus, for example, hemodialysis extracellular vesicles. So that's why we focused mainly on the in vitro analyses. So those are the ones, yeah, where we actually did tube formation assay and migration assay, at least on primary endothelial cell lines. And here we see the impaired migration, the impaired tube formation, and also the impaired proliferation that is induced. But of course, the message from these experiments are definitively limited. I agree.
Well, it is a very, but you don't know how many it's going to take.
Sorry?
You don't know how many EVs are going to be important in functional changes.
Yeah.
I admit, what about turning to analogous adult patients where you can get a lot of EVs and get some indication of functional changes?
Yeah. Yeah, so the problem with adults is we run into an hen-egg problem here because actually the lifestyle-related factors play a big role. Yeah, so we don't know exactly. So, for example, hypertension and diabetes by itself induce the release of extracellular vesicles. Yeah. So to really make a difference between what is first, what second is very difficult. Yeah, with the
these
use.
are great difficulties you're dealing with.
Yeah, unfortunately. So we really had a long discussions. Yeah, what we can do, what we don't or cannot do. I think really the most realistic way would be to pool plasma from the kids and then see how they work in vivo. Sorry. Yeah.
I agree. That would be a wonderful approach. Thank you very much.
Thank you very much.
All right, we have another question in the chat from Benedetta. It says, your data suggests that EVs are not just result of higher cardiovascular risk, but rather inducers. So would you suggest that EV removal during dialysis could be of benefit?
Yeah, definitively. So that's what we suggest. But the problem is that hemodialysis itself may cause the release of the extracellular vesicles. Yeah, so it is, I think, difficult. Yeah, but if we could design a novel dialysis machine that has filter systems where actually extracellular vesicles may be removed, then this could be a way to filter out the potentially harmful extracellular vesicles. However, I think if we filter them out, we may also filter out the good ones. Yeah, so I think not every extracellular vesicle is bad. Yeah, so and I don't know what consequences would result from extracellular vesicles that potentially are involved in repair mechanisms, and therefore I think we simply have to do more research actually to look at that. Yeah, but it's an interesting idea.
Yeah, one way in which this might be achieved is just the dialysis membrane itself, because our group looked a couple of years ago in HD, and we actually saw some reductions, and we were able to show that it was absorbing to certain dialysis membranes. So I wonder if just varying the dialysis membrane might have an impact.
Yeah, an alternative way, of course, would be the use of antagomers that actually would be supplied by nanoparticles, actually, in order to see if they could inhibit our specific microRNAs and therefore may reduce cardiovascular disease. And I think there were already approaches where actually antagomers were successfully used in patients to catch out specific microRNAs or disable their function in the patient. So this would be maybe a more specific approach to reduce the effect of endothelial extracellular vesicles. Yeah.
Yeah, I see Benedetta there. Did you want to follow up, Benedetta?
Yeah. Just a speculation on the fact that maybe also some toxins could be part of EV corona. So I really think that removing EVs, I don't know if it's possible a selective fraction or just a filter for maybe the majority of them. There really appears something to investigate more.
Yeah, definitely.
So I have another way out suggestion is with these small amounts of EVs involved, is it possible to inject them into these little vertebrate fish that have some sort of renal systems to get some clue about function?
Yeah, that's interesting. Yeah, actually, I have collaboration partners in Japan who do a lot of zebrafish studies on cardiovascular development, and yeah, of course, they would be maybe potential recipients where we need much less material. Yeah, that's a good idea, actually.
Well, Good luck.
Thank you.
You're very persistent, and you have to be.
There's one more question from Uta here, to compliment on the great data. Did you assess the vascular health of these children, and do they have already CVD evidence? Might be nice to correlate with your data.
Yeah, actually, so they show actually strong cardiovascular morbidity. So I just showed this at the beginning. So they all have, wait a minute, here we are. So they all show hypertension, yeah, and also actually some of them show even, so they all have left ventricular hypertrophy, and also they show resistant hypertension, yeah, although they received several multiple blood pressure medications, including a diuretic. And so actually, so they all show cardiovascular morbidity, and so this was actually removed in the KTX cohort, and yeah, so I think they have bad cardiovascular health actually.
What about BMI? I'm just looking at this, especially with the resistant hypertension in that population. Was there differences in BMI?
I would have to look that up again. So but actually I think I don't think it was statistically significant. Yeah.
Yep. Okay. Well, maybe we'll do a last call for questions. I think you've certainly generated some stimulating discussion today, Dr. Simmons. Really appreciate that, and really appreciate your being so game for the exchange. So.
Yeah,
Fantastic.
thank you very much for all the very interesting points and questions raised. I mean, I think it's really an interesting study cohort, yeah, to look at pediatric CKD patients, and I think they need more attention, although it's a very tiny group of patients. But I think as a model system to look for CKD pathophysiology and cardiovascular disease pathophysiology, they are really a good model system for us, yeah.
Yeah, I mean, I think the one takeaway is that everybody's really interested in where this goes next as well. So.
Yeah, let's see. Yeah.
Please join me in thanking Dr. Simmons for his presentation today.
Thank you very much.
Thank you very much.